Hello, everyone, and thanks for joining us today. I'm Dave Iverson, contributing editor at the Michael J. Fox Foundation for Parkinson's Research and the moderator of our continuing Third Thursday series of webinars on the latest in Parkinson's disease research. As you see on the slide that's on the screen today, our topic today is Parkinson's genetics and how that's impacting drug development. You also see a few tips on the screen, which are how to contend with problems that sometimes occur in any of these sorts of technical presentations. So you may want to just glance at those before we get going so that if something indeed does happen during our conversation today, you'll be able to contend with that in good shape. As always, you'll be able to submit your questions throughout the hour. You'll see a little Q&A box near the middle of your screen, and you can type your questions there. And we'll do our best to get to as many of those questions as we can. And as always, we're also providing these slides for download. There's a box called Resource Lists on your screen, and you can click on that, and then the document will open in a new browser window, and you'll be able to save or print from there. So let's get started, and let's talk about the kinds of topics that we'll be covering today. We're going to go over a real variety of topics that interact with genetics. There's so much interest right now in genetics in Parkinson's disease. And so we're going to talk about what genetic mutations are, first of all, and then we'll talk about what we know about that in terms of its impact on Parkinson's disease. We're also going to talk about a variety of new studies in genetics in Parkinson's, also how you can participate in those kinds of tests should you be interested in doing so. And we'll also explore what it means to get genetically testing. The genetics counselor is going to be part of our conversation today, and we think it's really important to get some sense of when should you consider testing, why, what are the considerations involved. So we'll go through that as well and talk about then the kinds of possibilities there are for genetics research. Let's go ahead and meet who's joining us today. We have Barbara and Jay Robinson joining us today. They're from Illinois, although today they're actually involved in participating in a genetics research project in New Haven, Connecticut. They both have family members with Parkinson's disease. And though they are both, thankfully, still free of Parkinson's, they are carriers of particular Parkinson's-associated genetic mutations, hence their great interest in this topic and in participating in genetics research. Barbara and Jay Robinson, thanks so much for being part of our conversation today.
Thank you, Dave.
Thank you, Dave. Our pleasure.
We're happy to have you join us. Joining us as well from Mount Sinai Beth Israel Medical Center in New York City is Dr. Rachel Saunders-Pullman, who is a associate professor of neurology at Mount Sinai and also is involved in genetics research as it relates to Parkinson's disease. Dr. Saunders-Pullman, thank you as well.
Oh, thank you.
And Deborah Raymond joins us too, also from Mount Sinai Beth Israel. Deborah is a genetics counselor, and we'll be talking in a bit about what a genetics counselor actually does and why that sort of expertise we think is so important when you become engaged in the question of genetic testing and participating in genetics research. Deborah Raymond, thanks for being part of our webinar today too.
My pleasure.
All right, let's first begin by talking some about what genetic variation actually is, what genes actually are, the role they play in determining who we really are. We have some 20,000 or more genes, each of us. And Deborah Raymond, give us a little sense of what genes actually do, because despite the fact that there's so much variation in our human population, we're really more alike than we're not. And yet it's genetics that can make those shifts and alterations and are responsible for our differences. Give us kind of a big picture view of what genes do.
Sure. Well, genes are really the manual of instructions of what makes us who we are. They determine, things like eye color, hair color, and all the different functions of our bodies and how we develop, and also have an impact on some of the different diseases we may have or other traits that we have.
Genes. Sorry to
Go ahead.
interrupt. I thought you had paused. I'm sorry. We see the question of disease in particular, which is obviously relevant for our conversation today, how is it that genetic mutations may impact the possibility of acquiring a particular condition? We see at the bottom of the screen this idea that genes are involved in how proteins are developed. What's that relationship, and how does that relate then to the particular question of propensity for a particular disease?
Well, while genes are the instruction manual, the protein is what the gene makes, and the protein is actually the functional part in the process. So the protein carries out the instructions of the genes. And when there is a mutation in a gene, that means there's some sort of change that occurs in the gene. And that change may be one that affects how the protein is made or how the protein functions, and whether it's able to do what it's supposed to do.
And it's that question of how proteins function and whether they're able to do what they're supposed to do at that very in the biological pathways that we think then plays a role in the possibility of disease?
Yes, exactly.
Well, let's talk then more about where we get these genes and how we're involved in getting the genes that we have. We see on the screen that we know, of course, that genes are inherited and that we get a copy. They come in pairs, and we get a copy from each parent. And again, as we consider this in connection, as we will in a moment, with Parkinson's disease, what's so important for us, Deborah Raymond, to understand about inheritance and the role that we have in getting pairs, so that it's important to understand how it's not just one copy, sometimes it's two copies. So why is that so crucial to our understanding of the role genetics play?
Well, because when there is our genes function in pairs as well, so that sometimes having a mutation in one copy of your pair of genes can predispose you to an illness, and sometimes you have to have a copy of a mutation in both copies of those genes coming from both parents in order to have a predisposition to disease. In those conditions where you only need one copy, that's called autosomal dominant inheritance, and that's what we see most often in Parkinson's disease. And the type where you need two copies is called autosomal recessive, and it's much more rare and seen in Parkinson's disease more rarely, usually in more early onset forms.
So there are some some gene mutations where you just need to get one copy from one of your parents, and that would put you perhaps at higher risk for Parkinson's. That's what happens more commonly. But in some rare cases, you'd have to get that gene mutation not only from one parent, you'd have to get it from both before you'd be at greater risk. Is that correct?
Yes.
Okay. All right. Thanks for that little bit of genetics 101 to get us started. Let's dig into then what this will mean in terms of Parkinson's disease specifically. So we see that there is a wide spectrum of risk with this question, as Deborah was just beginning to describe, and that some of those have a greater impact on the possibility of disease than others. So, Dr. Saunders-Pullman, let's bring you into our conversation at this point and give us a sense of some of these points that we see on the screen. Point one being that some mutations in and of themselves are enough to increase the Parkinson's disease risk. We'll talk in a moment about how genes may interplay with the environment to create risk, but there are some genes that just if you have that mutation, puts you at greater risk?
That's right. So the most common Parkinson's genes are associated with a single mutation, and these are LRRK2 and glucocerebrosidase, or GBA. There's also a much less common dominant gene, alpha-synuclein, where one only requires one hit. But it's really important to consider, if you have a mutation in LRRK2 or GBA, that you won't necessarily develop Parkinson's. And the group joining us today, I think, makes that very clear. And in fact, in the Michael J. Fox LRRK2 Ashkenazi Jewish Consortium, our group found that 70% of people with a LRRK2 mutation did not develop Parkinson's by the age of 80. So we do believe, as you mentioned, that other factors, including other genes, environment, are also playing a role, either in helping to cause Parkinson's or also in protecting us against Parkinson's.
We'll hear from Barbara and Jay Robinson in just a moment about their particular situation, but it sounds like you're saying that there may be you may carry, let's say, a particular mutation, like the LRRK2 mutation, which we'll get into more detail in just a moment, and just go through life swimmingly, just sail right along. And part of what we want to understand, Dr. Saunders-Pullman, is what might then be going on that would keep that person from getting Parkinson's, and then on the other hand, what might tip someone with that LRRK2 mutation towards getting Parkinson's. That's some of what we don't yet understand.
Exactly, and we think it's very important to understand, because if we knew protective factors, we could apply those potentially to people who already developed Parkinson's, or use them to decrease somebody's risk of eventually developing Parkinson's. And same thing, if they're actually deleterious factors, then we want to address those. So those would be things that would be more common in gene carriers with Parkinson's compared to gene carriers who didn't have it.
And what are some of those protective factors? We may not know them, I know, definitively at this point, but do we have an idea of, if someone is carrying a particular genetic mutation that puts them at risk, what it might be that would prevent them from going on to actually get the disease?
Well, it's very interesting. We have some, in Parkinson's in general, some things that are associated with lower risk of Parkinson's, such as smoking and caffeine intake, particularly for men. But we're still not sure what the role of those factors will be for LRRK2 and GBA. So I think that this is a really important area in research. Some of the other questions that come up, other things that may play a role in developing Parkinson's overall, calcium channel blockers, potentially non-steroidal anti-inflammatories, and then the other issues about any of the gut-related factors. We're not sure what role they play in LRRK2 and GBA, and these are, as I said before, very important factors in trying to understand both diseases and the progression and come up with better agents to treat them.
And a number of those things, of course, are being tested now. You mentioned calcium channel blockers. Those are drugs typically used to treat hypertension. There are clinical trials going on now to see whether or not those drugs in some ways are also preventative for Parkinson's disease. You mentioned smoking. Of course, we don't want to encourage people to smoke, but there is testing going on to see whether or not nicotine patches in some form might also serve that preventative purpose. So lots of interesting work being done to identify what some of those factors are that might prevent you, even if you are carrying a genetic mutation that increases your risk, might in the end be preventative. Let's talk a little bit about the next point, and Debbie Raymond, maybe you can comment on this, that mutations also may be linked to different symptoms in particular, or rates of progression. What are we learning about that?
Yeah, so basically the idea is that mutations have different effects on the protein. Some of them may impair the protein not at all. Some of them may impair them a little bit, and some may impair them a lot. And so you can see, differences sometimes when you do genetic studies in whether people who have a particular mutation in a gene share certain features such as, greater likelihood to have earlier or later onset. Maybe they have other associated features of the condition, more likely to have a balance issue or less likely, that kind of thing. So different mutations may act differently depending on, how they affect the function of the protein.
So this is, I think, giving us a good sense of the varied way in which genetic mutations impact both the risk of Parkinson's disease, but perhaps also those factors that may play a role in either lessening that risk or increasing it, in the way in which some of those genetic mutations may have an impact on particular symptom experience or age of onset. But obviously lots more to figure out about this. Let's dig into some of the specific genetic mutations that are involved in Parkinson's disease, and we see those up on the screen now. We mentioned the what's listed as the SNCA gene mutation, which has to do with alpha-synuclein itself. We'll come back to that, but let's start first with the two we've already talked about, LRRK2 and GBA. And let me use this moment to bring in Barbara and Jay Robinson, who we introduced at the beginning of the webinar, and who are people who have a particular interest in this. Barbara has two family members who have Parkinson's disease. Her mom had Parkinson's. She has a brother with Parkinson's. Jay has a father with Parkinson's. And for all of these reasons, they decided to participate in genetic testing and now in genetic research. And Barbara Robinson, why don't you first describe, if you would please, what you discovered when you did do genetic testing.
So I discovered that I carry both the GBA gene mutation and the LRRK2 gene mutation.
And Jay, how about you?
I discovered that I'm the carrier for the GBA.
And so you elected. Go ahead, Jay.
I was going to say, I'm not too surprised simply because my father had it, and I figured that there was always a possibility that I could, and that's really one of the main reasons I wanted to be tested to begin with.
And so you made this decision to participate not only in get genetically tested, but to also now participate in genetics research. In fact, you're in New Haven, Connecticut today, participating in a genetics research project sponsored in part by the Michael J. Fox Foundation called the Parkinson's Progressive Markers Initiative, to try to determine what's going on with genetic mutations in part of the reason for the study, and whether or not some of those things can help us get an early lead on what goes on in the development of Parkinson's through a better understanding of what's called biomarkers. And Deborah Raymond, before we go further with this is a good point, I think, to also introduce the role of what you do as a genetics counselor. We'll talk more about some of the considerations in genetic testing. But why is it important for people like Barbara and Jay and others who are now participating in these studies to have some sense of what may be in store for them, and what is it that a genetics counselor in particular can do in that regard?
Sure. Well, genetic counseling is really an opportunity for people to explore what is known about the genes for Parkinson's disease and what is not known. And, what is based on their specific family and sometimes their ethnicity, we can have a discussion about what the most likely pattern of inheritance is in the family, what are the risks to other family members, and also is genetic testing something that people think overall is something they would like to do. Would they like to know whether they carry these genes or not? And there are, people feel differently on both sides of this. A lot of people want to have, some insight about what this information might do in terms of If they have Parkinson's, does that change their medical management? If they don't have Parkinson's, is there something you can do to prevent it? Are there opportunities to participate in research or in other positive, aspects like potentially clinical trials? Some people feel that that's an important piece for them. Some people, think that, let's wait a little bit and see if things change over time, and maybe then, the decision to test changes. So there are different considerations regarding, how this information will impact yourself and your family, because it is genetic information, and how you kind of see it in terms of the benefits and potential, disadvantages as well of testing.
We'll get into this in a little more detail later on, but while we're on the topic, Barbara Robinson, maybe you can explain why you did go ahead and make the decision to not only get tested, but get involved in genetics research.
The initial interest in finding out the status, just to do the testing in the first place, came as a push from my family as a whole. My brother, who has the disease, was going to get tested. My sister has been doing fundraising programs for Parkinson's disease, and she right away wanted to get tested. She asked me, had I gotten tested? And I said, and I didn't even know anything about it, and so they both encouraged me to do it. And our family has a science basis and has experience with research projects in the past, and so we were all very interested in knowing our gene status. And so I am one of 4 children, and 3 of us were immediately tested because we just wanted to know where things stood. And my younger brother has no interest in testing because he doesn't see how it's going to change anything for him at this point.
And that's an interesting point, because I know as a physician, Barbara Robinson, you also happen to work with patients on this, and you have, I know, an interesting perspective on this question about when it makes sense to get tested. Why don't you add that, if you would?
So one thing that I also discuss with my patients regarding any test is that a test shouldn't really be ordered or performed unless it's going to change what you do in some way, shape, or form. And otherwise, there's no point. Sometimes it's very interesting, but it may be a waste of time and money if it's not going to change anything. And so along with the testing that we opted to do here was the sense that, and people ask me this all the time, well, so how is this going to change anything? You're positive for 2 genes. How is the research that you're participating in going to change anything? And my response is that hopefully it'll change something for me, but even if it doesn't, we need to collect the data and have the information to help potentially ourselves, and certainly to help other people and maybe even our children who may be at increased risk of Parkinson's or other diseases. And if you don't collect data, you will never get any information. And so that's how it's going to change my management in this case, is I'll participate in research. So therefore, it's important to have the information.
It's a great point, I think, Dr. Saunders-Pullman, and I'm sure it's something you work with when you talk with patients, as of course does Deborah Raymond as a genetics counselor, in that people shouldn't have the idea that if they learn they have a gene, there's a fix for it. We're not at that point yet. The point of participating in research is not so much that there will be a pill on the other side of it for you, so much as there will be data on the other side of this, and that it's that data that we need if we're going to develop a greater understanding of these particular genetic risks. Is that how you view it as well?
I think it's definitely important to think about, what is actionable, and we actually are getting to the point, though, where there are trials for GBA-related mutations that are moving forward. So we have to, if the algorithm is shifting a little bit, we're starting to think about things a little bit differently. But I also wanted to get back to the point about the type of research that people are engaged in. Some studies do require you to find out what your gene status is, and in other studies, you're not required to find out whether you harbor a certain mutation or not. And that can matter sometimes as far as some of my patients don't want to burden their children with an increased sense that they have a genetic form of Parkinson's. Others feel strongly that they want to know all the information they can. So it really is a very individual decision. I think that we're not at the level where if I know that somebody has a mutation, I'm going to give them a different treatment. We hope, though, that down the road we will be able to prescribe different treatments, and there are some important questions that we haven't resolved that we think are very important that we're getting in the direction of understanding. For example, even the referral for deep brain stimulation therapy, whether people with a certain genetic mutation might respond better. So these are questions that really are, they're not just for down the road, they're happening now. But as I said also, it's a very exciting time because we're on the cusp where some of these trials are also beginning.
Yeah, that's a really interesting point, that we're in this dynamic place right now where we're getting closer to a deeper understanding of how some of these mutations may have a direct impact on particular treatment forms, and also, as you suggest, getting to the point where we may be able to begin to treat some of these genetic mutations, which of course tips the balance very greatly in terms of what people may want to choose to do. We'll talk more about the genetic testing question in a moment, but let's spend a little bit more time on these specific mutations. We've heard that Barbara and Jay Robinson each carried the GBA gene, and Barbara also carries the LRRK2 gene. So let's talk a little bit more about LRRK2 in particular, because it's the most common genetic mutation that can lead to the potential of Parkinson's disease. Deborah Raymond, I'm not sure either of you, Deborah Raymond or Dr. Saunders-Pullman, can take up this question, whoever you feel is best to do so. But tell us a little bit more about the LRRK2 mutation in particular, what degree of risk it carries if you happen to have that mutation, and what we know about the demographics of it. We see that it's particularly pronounced in a couple of demographic groups, but where else do we find it too?
Yeah, so I think with LRRK2, we need to think about, first of all, it's leucine-rich repeat kinase is the protein. And with LRRK2 mutations, as I mentioned, only about 70% of people in our group, which was a cohort of Ashkenazi Jews from the U.S. and from Israel, only about 70% developed Parkinson's by the age of 80. LRRK2, I'm sorry, forgive me, 70% didn't develop Parkinson's by the age of 80. So only about 30% actually do develop Parkinson's by the age of 80. And I think there is the LRRK2, the Parkinson's associated with LRRK2 is very similar to other forms of Parkinson's. There are some group differences, but these are not very large differences. Some question about whether it may be a more slowly progressive form of Parkinson's, and also whether there's more issues as far as walking and gait, but without cognitive problems that are often associated with walking and gait problems. LRRK2, so I mentioned in the Ashkenazi population, there is one particular mutation that is much more frequent, and this is the one that's really most frequent in North America as well. We've also found, with a very small study, it was increased in Puerto Ricans. There's a group in Norway with a higher rate of this particular mutation. And then in the Basque Country, there's a different LRRK2 mutation where there is also a founder effect, and these founder effects generally arise from bottlenecks in populations where a population is concentrated in a certain area, so the genes that are more common in those ancestors tend to get passed along, and as those ancestors have many children and grandchildren and so forth, then it's more common in a particular group. So even though it's dominant, it's more common in a group, so you just need to have one copy.
And as we look at these particular three that are identified, it's also important, Deborah Raymond and Dr. Saunders-Pullman, to realize that these are just three of more than a dozen genetic mutations that have been identified as connected to Parkinson's. There are many. We've singled these out because this is where sort of the heart of the research is right now. But there's lots more to know, right? I mean, we're just at the beginnings in some ways of our understanding of genetics and Parkinson's. It wasn't so long ago that we didn't think there were any genetics at all. So it wouldn't be surprising, would it not, Debbie Raymond, if we learned that, you know, tomorrow there was yet another genetic mutation involved in Parkinson's?
Of course, yeah, and we expect to learn that there are more genes over time, for sure.
And I just to put a plug in that even if people don't have LAR2 or GBA mutations or some of the other mutations we didn't talk about associated with young onset Parkinson's, particularly the recessive ones, Parkin, DJ1, PINK1, that people still do sometimes have family histories of Parkinson's where we then presume there is a strong genetic component and don't have any of these genes, and we're looking to find other genes. I agree that we certainly think there will be new genes, and then as we've heard about today, sometimes people can have two genes, even two dominant genes.
So let's talk about where we hope this research will go in terms of how it may lead to specific treatments. And then we're going to also return to the topic of genetic testing. I know there are lots of people writing in questions now wondering about what they should do if they have a relative who has PD and whether or not that means they should get tested. I promise we'll get to those questions and take all of your questions about that in just a moment. But we want to spend a little more time first about why we think genetics is so important, even though it represents a relatively, what is it, Debbie Raymond, maybe we think 5 to 10% of Parkinson's has at this point a direct genetics connection. Is that about right?
Yeah, that's about right, maybe a little bit more.
Okay.
And when you say when you say a direct genetic connection, you mean one of these, what we call a major gene that we can say, this increases your risk. But we should probably also mention that there's a lot of research going on right now about risk factors and their relationship to Parkinson's, and these are sort of more diffuse genetic components that may increase or decrease your risk just a tiny bit and contribute to whether you will develop Parkinson's as well. So while yes, there are a relatively small percentage of cases that we can attribute now to being genetic, we do think that there are a lot of other genetic components that are brought into play.
And will no doubt be more and more of those as we go on. If we were to have this conversation 10 years from now, I suspect our percentages for the amount that we think that genetics influences the possibility of Parkinson's would be that much greater. But in the meantime, we want to talk some also about how genetics may lead to specific treatment possibilities. And the first is what we see up on the screen. And Dr. Saunders-Pullman, maybe you can describe a little bit where we hope to go. You were saying a bit ago that we hope we're getting to the point where we may actually be able to fix some of those dysfunctions. What is it that would be involved in that? Can you give us just a little glimpse into where you hope this research will go? Is a gene mutation fixable, correctable? How would that actually lead to a specific treatment for a genetic mutation?
Yeah, so I think maybe if you don't mind just to step back a little bit to say that, even where we don't have a specific fix, as in at this point with alpha-synuclein, which is a pretty uncommon mutation, that understanding the genetics really has been important in unraveling what's been going on in Parkinson's and identifying causes and targets. And this is really the great story of alpha-synuclein, where very few people have this, but it's really revolutionized Parkinson's research. And in this regard, some of the vaccine work that's going on with Parkinson's is directed to alpha-synuclein. So we're not fixing the actual mutation, but we're understanding what was causing the Parkinson's, and it's really understanding the genetics that led us to understanding Parkinson's better that has led to better treatments. And then the next step is, how can we be really directed as far as if we know that somebody has a particular mutation, can we treat that particular disease? And this also has a similar corollary to the synuclein story in that some of the medicines where the medicines are being developed based on a mechanism that was discovered through genetics. So I think if we think about GBA, I think that there really one of the big things is that with GBA, we understand a lot more than we do about some of the other conditions, a little more than LRRK2, because GBA mutations, when people carry two of them, cause Gaucher disease. So even before a link to Parkinson's was made, there was work going on in GBA, and there are different ways, as Debbie talked about before, the fact that we're identifying proteins and we know what these proteins do. With GBA, we don't know exactly how they're related to development of Parkinson's, but we have some pretty strong data. And so there are different approaches with GBA. So when we think about this protein, we think about an enzyme that has something called a substrate, that the enzyme helps break down the substrate. And with GBA, one of the concerns is that there is too much of the substrate, and that's backing up and causing problems. Another way to approach it is to actually increase the activity of the enzyme, or to go on a gene therapy approach and increase the enzyme, increase expression of the enzyme. And there's one study that has already been posted and is enrolling, and this is on the Clinical Trial Finder website for a substrate reduction therapy. And this is just in its early phases. And then there's some other medicines that are also, one being tested is a very interesting compound called Ambroxol, which may actually help the function of the enzyme. And this is fascinating because at low doses, it's actually an over-the-counter medicine to help thin mucus. It will require, we think, much higher doses as far as potentially treating Parkinson's. And there is a study in Ontario which has also been posted that's not looking just at GBA carriers, but at people with Parkinson's disease and dementia, and trying very high-dose Ambroxol. And then there is a company in Boston, LTI, that's focused on research development of a medication to affect GBA, and I understand it's in preclinical development now, and they may begin safety studies within this year as well. So there are a lot of different things going on, and both approaching the gene directly as well as really understanding how the gene works and developing medicines around that. And I think that one of the questions is, well, how do we modulate the gene? And it's not Parkinson's related, but in the neurology community, there is a very exciting development as far as a medicine for spinal muscular atrophy that actually increases expression of one of the compounds that's a problem. And it's just very exciting that in one area when we have a tremendous development and that this can often carry over to other fields. So it's an exciting time.
Yes, and I think that global look that you gave us of all of this points to the various ways in which progress is being made. Some are very specific, like the GBA drug trials that are beginning now, and the one in yet another neurological condition that you just mentioned that may lead to specific fixes for specific mutations. But I do want to underline the point you made that I brought up on the screen while you were talking, that bullet point, that part of what is useful about this genetic research is that it gives us a greater understanding of what goes wrong in Parkinson's for everyone, not just for people who have genetic mutations. And alpha-synuclein, of course, is the key point here. There are very, very few people that carry that mutation that causes too much alpha-synuclein expression that leads to the clumping up of that protein in the brain and kills brain cells. That happens very rarely, but we know that those clumps of alpha-synuclein occur in everyone with Parkinson's. So just by understanding this one small, tiny population, it's given us this broader understanding of the disease and may lead to broader applications that will be important for everyone with Parkinson's. Let's move on, though, to our I think the question that I see coming up the most on my screen from people, which is about how to get involved in genetic testing and genetic research. And we want to spend the rest of our hour focusing on those questions because there are so many of them. And let's just go through them as we can. First, a question for you, Debbie Raymond. Daniel writes in, how do you get a genetics counselor?
Well, luckily, they're pretty easy to find. You can, any major medical center has a genetics department with genetic counselors available. There are some that specialize in movement disorders or Parkinson's disease, and there's actually a website at the National Society of Genetic Counselors that has a referral listing as well, where you can choose by specialty or geographic location as well. So pretty easy to find. Your doctor can probably also tell you where to find a genetic counselor in your medical center as well.
And would you mention that web referral process again? Where is it that you can go that will tell you where you can go about finding a counselor for specific diseases?
It's called the National Society of Genetic Counselors.
Great. Okay. I'm sure people can Google that and go from there. Lots of people are also just writing in and saying, I have a parent, I have a sibling, I have an uncle or an aunt who has Parkinson's, and sort of what does that mean for me? What is the risk for developing the disease? I know that's a hard question to answer, Deborah Raymond, because it obviously depends. We don't really know. But if you have a relative with Parkinson's disease, let's start with a parent, and you haven't yet gotten genetically testing, do we know that necessarily increases your risk for Parkinson's just because you have a direct blood relation who has it, even if you don't know what the genetic status is?
Yeah, well, we think that does increase your risk. And if you have a parent with Parkinson's disease, your risk of developing the disease is probably on the order of 3%, 3 to 7%. So still, relatively low, but there is an increased chance of having the disease as well if you have a parent with it.
And let's bring Barbara and Jay Robinson back into our conversation at this point, because I'd be interested in hearing from both of you about how you've dealt with that question of risk in your life. Jay, you have a father who had Parkinson's. Barbara, you have a mother who had Parkinson's, and also a brother. You're also parents, so you think about this, I know, in terms of your sons. Curious about how you deal with that question of risk in your own lives, and what you'd say to others who may be scratching their heads, who are in similar circumstances, and wondering about what the right thing to do is as far as getting tested or participating in research. Barbara or Jay?
Well, for me, when I saw what my father was like when he had Parkinson's and leading up to the time that he passed, it made me aware that because he was my father, that this could be something that I would have to deal with someday. However, at the same time, I didn't press the panic button. I wanted to be educated more about it, and when given the opportunity, participate in the research study. I think that it would be wise for anybody when they know that they might be susceptible to something like Parkinson's. Learn as much as you can about it and get involved if you want to be more aware of what could be happening or may not be happening down the line. I have a twin sister who I used to wonder since I was tested and found to be a carrier. I've since learned that just because we're twins does not necessarily mean that she is also a carrier. And I think that there are probably other people in that situation, whether it's a twin or just a sibling. My sister or my brother probably still has it too. But through education, I think it clears up a lot of questions and it helps to prepare you for what may or may not be coming down the line.
It's a great point. And Barbara, would you concur that just one of the things that's helpful about getting engaged in research is you just get that much smarter about your own understanding of risk? I remember a friend of mine saying once that, if Americans really understood odds and risk, no one would ever play the lottery. And so one potential advantage, I guess, of getting involved in this is you just get smarter about what the real risk considerations are.
Absolutely. I know that with 2 family members with Parkinson's, I feel like the risks are astronomically high that I could get the disease. But as we get more information, even having 2 mutations, as Dr. Saunders-Pullman pointed out, my risk is probably in the 30% range, not in the 50 to 80 or 90% like I might have been afraid it would be. And so knowledge is power, and it gives you the ability to have power to try and collect data and change things, and also the power to have a better outlook, in my opinion.
And actually, I have a comment about the knowledge is power point.
Go ahead.
So I completely agree with that, but I just wanted to put in a little caveat there that you also have to be careful about what knowledge you internalize, because a lot of this stuff is so new and is still being figured out, and a lot of the things that Barbara was just talking about have been proven and reproven, basically. But there are other things. For example, the chance that you with LRRK2, the chance of having Parkinson's if you have the gene has been studied by many, many different groups, and there was one very large study that showed the chance of having Parkinson's if you have a LRRK2 mutation by age 80 was quite high, and many other studies have since shown it to be closer to that 30% mark. And it's not that the first study necessarily had anything. The data was false. It's just that sometimes there's different biases that are introduced, or they may have been looking at specifically familial cases that may have been more likely to show signs. So you do have to be a little bit careful with the information and realize that we're still learning so much and that it's going to change over time.
And I think something to add to that. Yeah, it's just the issue about the meaning of a negative test. So it's very important what one gets screened for. So if you're of Ashkenazi background, then there's certain GBA mutations that are certainly of greater risk, but a lot of the studies only look at the one mild mutation, N370S. So just because you're negative doesn't necessarily mean you don't have any of those other mutations, or so it's just important to stay on top of the knowledge and to realize that you are gaining information and power to deal with these things, but we also have to realize there is still a lot of unknown as well, and it's important that we keep that in the consideration too, as far as that the data keeps improving, and we're understanding more and different things than we did before.
Let me follow up on that with another. Yeah, go ahead, Barbara.
I was going to say that speaking from the participant in research and the test subject and the the patient point of view, which is not always my point of view, it is kind of a roller coaster ride. You get one piece of news and it's like, oh no, and then you get another piece of news, well, oh good, and then another piece of news, oh no. And the key, I think, to dealing with all of this, at least for me, is to remember that the only way to really figure out what's real and what's not and what's going to be helpful is to collect the data and have the knowledge. And so I'm just holding on for the roller coaster ride and just keep saying, let's just get that data, one foot in front of the other, keep doing it, and in the end, things will wash out and be more clear.
Thanks for adding that perspective. Really, really useful. Let's pursue this just a bit more with a question that a lot of people are asking that has to do with what we know and what we don't know. And this specifically, Debbie Raymond, has to do with, I think, some uncertainty from people about what you actually really can learn from a genetic test, what it can tell you and what it can't tell you. For example, we're having a number of people writing in saying, if a genetic test will tell you not only whether or not you, may have a genetic mutation for PD, but then how fast your Parkinson's would progress or what specific treatment you should follow. As Dr.
Right.
Saunders-Pullman was saying earlier, we're getting a little closer towards maybe it having some impact on treatment options. But at this point, when you take a genetic test, and let's say you learn you have the LRRK2 mutation, what does that tell you and what doesn't it tell you about what kind of PD, if I can use that phrase, you might get, how fast it might progress, or what your treatment options might be? What is it telling you and what is it not yet able to tell you?
So when somebody has testing, first of all, there's one distinction, which is that some people who have testing already have PD, and I'm guessing that's the group you're talking about. And then there's the group that doesn't have PD. So the testing experience is different, and the concerns may be different based on whether you have the condition or not. There's, I think Dr. Saunders-Pullman really kind of went over for LRRK2 the sort of differences that we can say now about how your PD might be over time. I think we think that LRRK2 PD may not progress as quickly as some, but I think I'm going to let her continue this because yes.
Yeah, well, I think that the issue is at this point what we're seeing as a group overall gives us some features and sense about prediction, but unfortunately none of this is predictive on an individual basis at this point. So by knowing that somebody has a LRRK2 G2019S mutation, I couldn't say that they would progress to this much faster, or that they would develop this kind of symptom at this time point. Even when there are mutations that are associated with cognitive decline, not everybody gets it. So I think that it's tricky because the kind of information we're gaining at this point, unless one's enrolled in a specific trial that's trying a new agent, individually isn't going to change your therapy or your doctor's giving you a prognosis at this point. We're hoping that eventually with precision medicine we will get to that point, but I don't think the expectation from testing for somebody who already has Parkinson's is that it will give you the crystal ball. Unfortunately, we aren't at that point. I don't know when we'll be at that point because, as we talked about, even developing Parkinson's, there's so many factors that play a role, and we need to separate out exactly which those other factors are, both things that are negative factors and also positive factors. And when I see somebody in the office, I don't know if they have these other protective factors. So there's a lot still not known, and I hope I'm very optimistic about this, and I study this because I think it's really important and will change the way we approach Parkinson's and treat Parkinson's and hopefully lead to real disease-modifying therapies. But I think we also have to step back and say there's only so much we know so far, and by getting the test at this point, it doesn't change whether I pursue a particular therapy or not. My hope is that it will change my recommendation in the future, and hopefully in the near future.
And then let's move to how one can get involved in Parkinson's research, should you be so inclined. And we've put up on the screen here a number of ways in which you can go about doing that. The Parker's Progression Markers Initiative is the first one, and Barbara and Jay Robinson are Exhibit A of the decision to participate in that. And Barbara, Jay, if you would, again, just make your observations known, if you would, about why you did make that choice, because I'm sure a lot of people right now are looking at this thinking about, well, I could do that, I could pursue one of these other trials. What was it in the end? You've both commented on this already, but I think it's worth underlining as we wrap up our webinar today, why you made the choice to get involved in that particular form of genetics research.
Well, while I've talked about my father having had it, I've been living most of my life, I'm married to a woman whose family has had it, watching how it affected her mother, who was a very strong and incredible person, and to see the disease tear her down bothered me a great deal. To see her brother, who was a high school classmate of mine, to see what it does to him and how it affects the whole family, motivated me to want to be involved at some level. When again, it's a matter of it hitting home, and if you are the ilk that you want to do something that maybe can be of help, you do something like participating in the programs that are available. PPMI, her sister, she told you, was a big factor in us getting in. But when it just kind of hits home, and like I said, I just wanted to be able to do something that might help somewhere down the road.
Well, thank you for making that choice and for being in New Haven today and acting out that choice in a very direct way. We also have up on the screen Fox Insight, which is something you can consider doing online, which is just something I participate in. It's a survey that you take a number of times a year, which tracks how you are doing with your Parkinson's diagnosis and allows a large collection of data to proceed about what's going on with Parkinson's, including the question of genetics. And it speaks to the point we made earlier that there's a way of participating in research and genetics research without necessarily having to learn what your own specific mutation is. And then, Deborah Raymond or Dr. Saunders Pullman, if you would comment on the last point, which is about enrolling in other kinds of Parkinson's genetics research. You can use Fox Trial Finder as one way to do it. There are others, but there are lots of trials out there, including the GBA trial you referenced earlier. What would you like to say in terms of asking people to consider at least engaging in that possibility?
I think that, as we talked about before, being informed as possible. And what's nice is the resource from Fox Trial Finder, which draws from the clinicaltrials.gov, does show some of the trials so that you can find out which are participating sites. Not all studies post on the clinicaltrials.gov, and this is an impetus to me to make sure we post as far as making sure our studies are on there as well. And I think to ask your neurologist if there's also something that a study that's happening nearby to become involved in, I think also is a good thing. But there are tremendous resources online, which is great in that way. I also wanted to just add for a moment, Debbie had mentioned the Genetic Counselors website, and that is actually nsgc.org, so National Society of Genetic Counselors.org, just if anybody was interested in that.
Great.
Thank you.
And Dr. Saunders-Pullman, you mentioned earlier, of course, the GBA trial that you're involved with, which is, I think, the first time that we're actually testing a potential treatment for the genetic mutation in Parkinson's. But we're just starting down that road, right? I mean, it would be your hope, I'm sure, that within the next several years that we will be testing potential treatments for other genetic mutations in the not-too-distant future as well.
Yes, that's certainly my hope. So I'm actually not directly involved in the trial yet. I'm actually doing a lot of work trying to understand GBA still further. But we're very hopeful for LARC2, but also to determine other mutations and trials related to them. And then the point you brought up before, Dave, is that even though these studies may start related to a particular gene, we really hope they'll be relevant to other groups of Parkinson's as well. So we're excited about that.
Well, thank you. Thank you all for the work that you're doing and for your participation in research. You all are engaged in that. Barbara and Jay Robinson, of course, along with Deborah Raymond and Rachel Saunders-Pullman, and we really appreciate both your participation in this webinar today, but also for the direct role you're playing in furthering our understanding of genetics and Parkinson's disease in general. Thanks all very much for your participation. We appreciate it, and we want to also mention that for our next webinar on the third Thursday of March, which is March the 16th, we're going to replay a webinar from our archive. It's the one on complementary and alternative medicine. It's an intriguing topic. We think you'll enjoy having a chance to participate in that. Again, well, it is one from our archive, and so there won't be a live guests. We will have staff standing by to still take your questions. So if you have questions about alternative and complementary approaches to treating Parkinson's, you'll still be able to get those questions answered. That is on March the 16th. Thanks again for your participation. I'm Dave Iverson.